Hey everyone, I hope you're all doing great. Well, in this video lecture, I thought of coming up with something very unique and like which will help you in the exam series. So I thought of coming up with a typical video in which I'll be discussing about just the topic, but from the point of view of an examiner. So what I want to say is, in every video, we'll try to analyze a topic from multi-dimension, and that will make sure that you get full marks out of that particular topic if it is asked in somewhere or the other. So for the time being, I thought of making this series especially for the AS and native biology. Very soon I'm going to come up with the same series for the IGCSE biology also, right? So let's begin with today's topic. So before that, let me, yeah, I hope that's visible. Correct. Now let me take the pen. Yeah. So today's topic is cell signaling. Well, now before talk about the cell signaling, let's understand. I have taken the question from cell signaling from these past years. There can be multiple, I know, but for the time being, for you to understand, I have taken 1, 2, 3, 4, 5 different questions which are already asked in the past years. So 2018 3 times and 2019 2 times. Even I have written the month, the series and the paper variant. Everything is written. I hope that makes sense to you. So now let's begin with the cell signaling as a mechanism. So what is the cell signaling? Well, cell signaling is a process in which a cell communicates with another cell. So basically what happens in order to communicate a typical cell will release some chemicals, and those chemicals will be received by the other cell. Let's give an example. Suppose you on the other side of the screen and me on this side. Say if we tend to communicate with each other, so what we'll do, either of the way, some chemicals will be released. Either it will be released by you or it will be released by me. So the chemicals are released by you. Now, if I want to communicate with you, those chemicals should be received by my end, right? So the moment I receive those chemicals, I will get your message, the message that you're going to deliver to me. And this way, you can communicate with me. I hope that makes sense. Now understand what all things I have said. Number one, in order to communicate, we need to have two cells. So for the time being, you have, this situation has you and me. So I've got two cells. So a minimum requirement is two. We got that. Second thing, there will be one cell which will be releasing some message in the form of chemicals, right? And there will be the other cell which will be receiving those chemicals. So for this particular examples, it's you who are going to send some chemicals to me. So the moment you receive those chemicals, I have to make sure that I grab those chemicals in order to communicate with you. Now, how do I grab those chemicals? Well, I have hands. So the moment you release those chemicals, I'm going to catch those chemicals. That means if you consider me as a cell and if you consider this is my hand, so this hand is my receptor, which will receive the chemicals which is coming from you, right? So let's quickly look on the screen here. Say this is you and your name is A, and this is me, my name is B, and say you try to communicate with me. So what you did, you released some chemicals here, right? So now what is my task? I have to make sure that I'm receiving those chemicals. Then only I can communicate with you, right? So for that to happen, I have to make sure that I have the hands. So say these are the hands. And the technical term, the clinical term of these hands of mine is receptor. Make sense? So with the help of receptor, I'm going to catch those molecules which are coming from you, right? So the moment I catch that, there will be an initiation of the cell signaling as a process. And that's how it occurs actually. And there will be a number of steps that we're going to see very soon. But hey, understand one thing. You want to communicate with me or the other way, one cell will synthesize or will release some chemical molecule which will be picked up by the other cell, right? So what all things you need to understand here? First thing is the identification of the cell, the identification of the cell signaling molecule, the identification of the receptor, and the identification of the target cell, right? Now this cell signaling molecule here, they are alternatively known as ligand. Make sense? So the moment you release the ligand from your end, I will make sure that with my hand, which is known as receptor, I will pick up those ligand and I will start communicating with you, right? But understand one thing. If something is coming onto my hand, there has to be a complementary binding, right? So that I can have a proper grip. This proper grip is known as complementary binding. So if you see, if I make them here like this, this is one of the ligand molecule. So what I can say here is, this particular situation will give rise to a complementary binding. Complementary binding. Complementary binding between who? Between the cell signaling molecule, which is alternatively known as ligand, and the hand which are known as the receptor. So there will be a complementary binding happening, right? So that's how it occurs. I'm not going to talk about cell signaling in details because the purpose of making this series is, if there is a question on cell signaling, what all points to be kept into the consideration so that you get 100% marks for the particular question, right? So just for your knowledge, the cell signaling can be of 3 different types: autocrine, paracrine, and endocrine. I'll give you a quick brief description about this. Autocrine means, auto means self. So that means if a cell is releasing some chemicals in the form of cell signaling molecules, and the same molecule will be received by the same cell. Auto means self, if you remember. So that kind of cell signaling is known as autocrine cell signaling. Paracrine. This is a classic example of paracrine. Why, sir? Because the distance between the two cells is not very far. It's a more normal, moderate, short distance cell signaling happening. This is an example of paracrine signaling. Endocrine signaling. When you hear this word endocrine, correct, we're talking about the hormones. So hormone means there will be one cell, I should say there will be the target cell very far away from the source. So if the cell signaling happening between two cells which are very far from each other, that will be known as endocrine signaling. So let's take an example. If this much is the distance, it is endocrine. If this is the distance, it's paracrine. If it's happening in the same cell, it's autocrine. Right? One more time. Autocrine, only one cell is involved. Paracrine, endocrine, more than a cell is involved. Our minimum requirement is 2. That's how the thing occurs. That's about the cell signaling, right? Now let's see, if a question comes on cell signaling, what all things you need to take into consideration, right? So the first thing, identification of the cell signaling molecule. Let me change the color once again, dear. Yeah. Identification of the cell signaling molecule. Well, in order to identify the cell signaling molecule, either some name will be given to you in the question paper. If there is no such name given, just write that chemicals. Just refer to that particular statement and you identify, you pick up the cell signaling molecule. That will give you 1 mark. The first mark will be given to you if you identify the cell signaling molecule correctly. Got the point? Second thing, identification of the target cell. Right? Let's take this example only. You release those chemicals and I grab those chemicals with the hands, which are known as receptor, right? So in this current scenario, I am the target cell and you are the source. Perfect? So if I am a target cell, that means I'm supposed to capture, I'm supposed to grab those cell signaling molecule which is coming from you. So in order to grab those molecules, I need to have receptor. So for me, third step will be the receptor. So if you see identification of the receptor, definitely they will be present on the target cell. Why they are required? Because unless and until you have this hand, you cannot capture this cell signaling molecule, right? And remember, if you make this cell, in most of the cell, the receptor will be embedded in the cell surface membrane. So this is a kind of embedding. So it will be embedded in the cell surface membrane, right? These 3 points, the moment you write, you get 3 out of 3. Next, like I have said, I can hold this pen because there is a complementary binding happening. As simple as that. So the moment it matches perfectly, that will give you the maximum efficiency. Same thing happens for the cell signaling procedure also. So there has to be one complementary binding to be taking place between the cell signaling molecule as well as the receptor. Next point. And what is that? This activation, this primary response, the moment you grab that primary response will initiate a chain of reaction and a cascade rather, which will involve the production of secondary messenger. Secondary messenger? Then where is primary messenger? Dear, you release those chemicals. I grab those chemicals with my hand known as receptor. So that communication, direct communication is known as primary messenger. The moment I get that, there will be some conformational change in those receptor molecule and subsequently following the cascade mechanism, a series of chemical reaction inside my body, because I am the one who is catching the molecules which is coming from you, inside my body, there will be the generation of secondary messenger, right? And finally, there will be an outcome and which will be definitely an internal specific response. So if you see how many points we have talked about here. So we talked about 1, 2, 3, 4, 5, 6. And not only we talked about 6 points, we talked about in a proper sequence. So guys, the moment you write all these points in your answer scheme, trust me, you're going to get full marks for that particular question. Right? Now can we take some questions? Yeah? Let's start. Okay. It's the first question. So I have already mentioned the year once again. So I'm not going to talk about that. It carries 4 marks. So what are the points you need to cover up? First, the activity of the SAN is controlled by the nervous system. Noradrenaline is released by the nerve cells in the SAN. Fine. Shows the role of noradrenaline in causing calcium ions to enter the cell. Guys, understand now. From the question I can understand, for me the final task means that internal response is this. I have to achieve this. I have to make sure that there is an entry of calcium ions happening inside the cell. So if at all I'm planning to develop a strategy for that particular cell signaling, I have to make sure that I am going to attain this function at the end. Right? Second, if you remember I told you first thing first, I have to identify the cell signaling molecule. So for me, these are the cell signaling molecules. The moment you identify them, you get 1 mark. Correct? Second, if you remember I told you identification of the target cell. So the activity of the SAN, right? So for me, SAN is the target cell. So the moment you do the correct identification, you get the 2nd mark. Third, if you remember, complementary binding happening between the receptor which is present on the target cell and the cell signaling molecule. The moment you write that, the receptor, the complementary binding, you get the next 1 mark. That is the third. Now understand, this binding is nothing but your primary response or primary messenger kind of thing. This will activate the production of CAMP. CAMP is a classic example of secondary messenger. If you see the name CAMP in your question paper, quote that. If there is no such name of a secondary messenger, no need to write any name, just write activation of secondary messenger. Since for me this question CAMP is given, what I can write activation of the CAMP. That will award me with 1 more mark. Right? And finally what will happen? This CAMP will make sure through the cascade that entry of calcium ion is happening in the cell. So for this particular question, what all points I took? Number 1, identification of the cell signaling molecule, that is noradrenaline. Number 2, identification of the target cell, that is SAN, cell, as per the picture. Number 3, the complementary binding happening between the receptor of SAN and the noradrenaline. Third mark. Fourth, this complementary binding will initiate the secondary response, which in turn will make sure that entry of calcium ion happens inside the cell. The moment you write all these points in the sequence, you're going to get full marks out of it. Guys, is it? Now the next question. I hope that makes sense. Okay, right. So that's a 3 marks question, alright. Following the liver tissue damage, chemicals are produced and released into the circulation. These chemicals are able to stimulate the liver cells to help tissue repair, alright. Explain how this is an example of cell signaling. Okay, guys, now understand. Will stimulate the liver cells to help tissue repairing. Now, repairing means they are actually talking about mitosis, right? So for me, mitosis is the final task to be achieved, right? So for that to happen, I have to initiate a cell signaling mechanism. Let's talk about it. Following the liver tissue damage, chemicals are produced. Okay, guys, this is your cell signaling molecule. But hey, where is the name of the cell signaling molecule? No need to worry. If the name is not given, do not write. Simply you write chemicals coming from the liver tissue damage is the cell signaling molecule. The moment you write, you get 1 mark. Point number 2. If you see what they're writing, these chemicals are able to stimulate the liver cell. Now understand one thing, guys. That stimulation can only take place if those chemicals are accepted by the liver cell. If I am the liver cell and if the chemicals are coming from your side, that means I have to take those chemicals. So what all I need to take? Those chemicals. But how? With the help of the hand. Hand means receptor. So your second point will be the target cell. And what is the target cell here? As you can see, the liver cell. Because you're going to stimulate the liver cell only. So liver cell is the target cell for you. Third, there will be a complementary binding happening between the chemicals and the receptor which is present where? Which is present on the target cell membrane. And that complementary binding will initiate the formation of secondary messenger, which in turn will instruct the cell to undergo repairing. That means mitosis. You write tissue repair, you'll get a mark. You write mitosis, you'll get a mark. It makes no difference. So again, guys, I have followed all the rules and regulations here, and I hope that makes sense. Identification of the cell signaling molecule. Here, no name was given, so I quoted chemical. Identification of the target cell. It's liver cell as per the question. Third, the receptor molecule. That means complementary binding happening between the receptor and those cell signaling molecules, which are alternatively known as ligand. Fourth, That will initiate a secondary response, I should say secondary messenger. And finally, my final thing will be the final task to be achieved, that is the mitosis. That means it will help in repairing the tissue. Guys, right? Okay, now next question. I hope you're getting a clear picture gradually. Oh, that's a 2 marks question, bro. Fine, let's see. Some epithelial cells in the kidney release the protein, vascular endothelial growth factor. Okay, so even before going through the whole question, I can say this is my cell signaling molecule. All right, it's already given. Cool. So the moment you write that, that will be your first point. This protein is cell molecule that stimulates cell division. It will stimulate the cell division in the endothelial cell. So for me, this is the target cell. So second point will be identification of the target cell. Right? Third, one more time, you have got the cell signaling molecule, you have got the target cell. Next will be the complementary binding happening. Complementary binding, pardon my handwriting please, between the receptor of the target cell and the cell signaling molecule. Fourth, initiation, the production of secondary messenger. Right? Guys, remember why I'm writing secondary messenger all the time, because the moment complementary binding happens, that itself is the primary messenger. This primary messenger will initiate some conformational change in the cell, in the target cell, and that will lead to the formation of secondary messenger. Please take that on a serious note. And the last point will be that is, it will stimulate the cell division, which is given in the question. So stimulation of endothelial cell to undergo cell division. You write all these points, you get a full marks. Correct? Okay, let's move on to the next question. Okay, again, it's a 4 marks question. Okay, cool. Platelets metabolize linoleic acid to produce a molecule known as thromboxane. Okay, so this is something important. Thromboxane is released by the platelets when blood loss occurs. They act on the smooth muscle in the wall of artery, and it will cause the artery to constrict, which will reduce the blood flow. Super. Now understand. Which will reduce the blood flow. So that means this is my final task. Right? This is where I want to reach. So I have identified my final task. That means I have already acquired 1 mark. Now let's start from the beginning. Thromboxane is my cell signaling molecule. 1 mark, done. Right? It's released by the platelets and it acts on the smooth muscle cells. So smooth muscle cells are my target cell. So 1 more mark. So I've identified cell signaling molecule. I have identified the target cell. Next, I will write about the complementary binding happening between the receptor which are present on the smooth muscle cell and the thromboxane. The third mark. Fourth, that is the initiation of the secondary messenger. And fifth, that is the final task, that is vasoconstriction. Understand, which reduces the blood flow, which will cause the arteries to constrict. That means the diameter of the lumen will become smaller. That is what process? Vasoconstriction. Correct, guys? I hope that makes very sense to you. You see, these are the questions. They're very easy. The moment you understand, the moment you crack the steps to write an answer, you'll get full marks out. Sorry. Yeah, that's the last question for the day. Okay, again, it's a 4 marks question. Let's see. The production of the milk by the cells of mammary glands involves an action of several different enzymes. Cell surface membrane contain a membrane protein PRLR. Okay. Shows an outline summary of HPRL movement in the production of breast milk. So for me, this is my final task. I have identified, that is production of the breast milk. So I have already secured 1 mark. Correct? Now if you focus here in the diagram, you see this particular cell is secreting these chemicals, these molecules, which are none other than your cell signaling molecule or ligand. So I have identified the cell signaling molecule. So I have acquired one more mark, right? Third, I have acquired one more mark just by identifying the target cell. That's the mammary gland cells. Because these are the cells which is to be stimulated for the production of the breast milk. Right? So I've achieved one more mark. Fourth, membrane protein PRLR. These are nothing but your receptor. So if you see a receptor, I already told you receptors are present on the target cell's membrane. So there is no exception for this as well. So here the receptors are present on the surface of the target cell and they are waiting to capture all those chemicals which is coming from the source cells. So the moment they come, there will be complementary binding happening that will lead to one more mark. After complementary binding, the next point will be initiation of the secondary messenger, correct? And your last point will be the final task. So this is your internal response. Understand that. Correct, guys? Fine. So I have taken 5 different questions from the past years, and I hope that the whole picture about the cell signaling is clear to you. Those who are studying in AS level, in the A level, there is a chapter on homeostasis. There you will be having a detailed discussion on the cell signaling. So that's all, guys. That's all for the day. I hope the video makes sense to you. Those who are watching my channel for the first time, do not forget to subscribe my channel. Give me a like, give me a thumbs up, and share the video with your near and dear ones. And write down in the comment section below whether you want me to continue this series for your betterment. And if you have some suggestions, do not forget, don't rather hesitate to write down in the comment section below. That's all, guys. Have a good day. Take care of yourself. Bye bye. Thank you.